On behalf of the members of the Academy, welcome to this episode of Learning and Living STEM in Connecticut, the podcast of the Connecticut Academy of Science and Engineering. My name is Tan Deleon. I'm an elected member of the Academy and serve as an officer for its governing council. For more information about the Academy, visit ctcase.org. Our topic today is accelerating the discovery of abiotic vesicles. Here to discuss what we should know about this and what is happening in Connecticut is Professor Sarah Maurer, Associate to the Dean and Professor of Chemistry and Biochemistry at Central Connecticut State University. She is also a member of the Academy. Welcome, Sarah. Can you tell us a bit about yourself?
Yeah, of course. So I am a professor of biochemistry at Central Connecticut State University, and we're a primarily undergraduate institution. So we have undergrad degrees in sciences, but also some master's programs that are really great. And I study biochemistry, but my focus is on lipid membranes. And like your title suggests, we can make lipid membranes out of things that are not biological, so abiotic materials. And these membranes self-assemble into structures that are sometimes called liposomes, or an alternative term is vesicles. And there's some nuances to when you use which one, but they kind of mean the same thing. We are interested in how these assemblies can act like living systems. So I really study the origins of life, or how life can form on other planets, and even thinking about like synthetic biology and how you can make life that looks very different from our existing Earth life.
Wow. Okay, that's pretty neat. That's really neat. So let's dive into this a bit. So I I think you kind of touched on it in your preamble, but why should the general public care about abiotic vesicles?
So there are lots of uses for these cell-like systems. A really good example of abiotic vesicles are the like lipid nanoparticle structures that we see for mRNA vaccines, like the COVID vaccine. But I apply this to a much more basic science, thinking about how self-assembly could help life get jump started. And I don't know how much everyone follows NASA missions, but we're actually sending a bunch of missions to places in the solar system where we think there might be life, or may have been life in the past. And lipids are very stable molecules, and so we expect that lipids would persist for much longer than other types of molecules like protein or DNA. And so it might be that we can discover things like lipid vesicles on Mars, in some of the waters that exist on Mars, or even in some of the subsurface oceans of the moons of Saturn and Jupiter. So we're sending a orbiter to Enceladus, and we're also sending a rover, an aerial rover to Titan, to look for these signs of life.
Oh man, so are you part of the whole exploration effort with NASA, or are you just providing research support? Like how does that work?
So I am not directly involved in mission planning, but I have participated in NASA grants that work to study these things. I've been externally funded by the NASA Exobiology program. I also have, gotten money for studying places like Titan and Enceladus through private funding. So there's lots of ways to assist with mission data by working to understand these processes without actually doing mission work. But if people are interested in like helping to plan missions, faculty members are definitely now involved in missions. And there was a really cool mission where we landed on an asteroid and brought material back from an asteroid that has never been, the material has never gone through the process of, the meteoritic process, so going through the Earth's atmosphere. So it's like pristine material, which we can now ask for as scientists, and we can analyze in our own labs.
Oh wow, yeah. So it hasn't been adulterated by all the
Yeah.
heat and everything. Wow, that's fantastic, really fantastic. So just for our listeners, when you're talking about lipid vesicles, you're literally talking about the fat vesicles, or are you talking about something more specific than that?
So lipids are a big class of compounds. When scientists were originally discovering biomolecules, they said, oh, there's these things that look like nucleic acids, and that's how they named them. And then the majority of our cells is protein, which we knew it had a very specific chemical signature. And then there was everything that went into a oil phase. So anything that would like dissolve in gasoline, essentially. They just kind of called a lipid. And so lipids are a big class of compounds. The lipids that form membranes are different than the lipids we use to store energy. So the lipids we store energy with are called fats or triglycerides. The lipids that we use to form membranes tend to have less oil in them and be more polar so that they interact a little bit more with water.
Ah, okay. Okay, yeah, thanks for that distinction. That definitely makes it a lot more clear now. So, so, so I guess what tools do you use to understand the origins of life?
Well, so the origins of life is a big term and it's a really cool interdisciplinary science. So there are geologists and physicists that work to understand how planets form and what molecules would be available from, for example, like mineral interactions, mineral water interactions, also how the atmospheres were. So we think about like atmospheric chemistry on early forming planets. But then also, origins of life chemists do things like look at existing organisms and try to work backwards in time to try to understand what early life looked like. So that's called the top down approach. And I am kind of in the middle where I am looking at um, biomolecules that we know have functions like lipid bilayers and trying to make molecules that are not biological do the same function. So when we think about lipids forming membranes, we know that they have kind of a two-part, they're amphiphilic. So part of them interacts with the water phase, that's the hydrophilic part, it likes water. And then the other part is this long hydrocarbon chain, which doesn't like water, and so it's hydrophobic. And this is how a bilayer self-assembles through the interaction with water, and it's an entropic effect. And we can make that happen in non-biological molecules. So we can use things like soap-like molecules to actually build out abiotic lipid membranes.
Okay, that's very interesting. Okay, so I guess what is the major challenge in understanding abiogenesis then?
So in my experience, the hardest part about understanding abiogenesis is that we only have one tree of life, right? All of biology is based on a single metabolism. We all use ATP synthase to generate energy. There's really only one DNA code. We use 4+1 nucleic acids to code for all of our proteins. We only have 20 proteins or 20 amino acids that we use to make all of our proteins. There's a central molecule, the ribosome, that connects these two polymer systems. And I think it's really hard to understand what a different form of life will look like, how to generalize rules for what is required for life when we only have one form of life. And there are some really interesting ideas for what weird life could look like. I know some people who say we already have weird life. It is what you see in every time you boot up a computer, you're generating in silico life, right? And so that's an example of weird life that we already have today. But maybe if you're a purist, you say, well, who made that computer? Is that just another branch of our tree of life? Because without humans, computers really wouldn't exist. AI wouldn't exist. And so is AI just a extension of the human genome? I think there are other instances, other ideas for weird life. For example, there are some people who believe that there is maybe they call it like dark life on the planet, or like a there's a form of life that we don't know about and we haven't recognized because it's so different than the other lives that we have observed. Um, and then, if we do go to Titan, for example, Titan has a very different atmosphere and temperature profile, and it is a very different environment for developing life. And so, what does life look like if instead of water, you are growing life in liquid ethane at a very cold temperature? And so, for me, I
Okay.
think it will be really hard to make any generalizations about abiogenesis until we see a second instance of a living system.
Okay. So yeah, so weird life. So I've never heard that term used that way before. So so you would consider, I mean, we'll talk PCs because most people have a computer. Uh, so some people consider. I just want to make sure I understand. So some people consider the computer booting up like silica life. Is that right?
Yeah,
Okay.
it's coming to life.
It's coming. Okay.
It's
All right.
taking in energy and it is processing information. Uh,
Okay.
in some cases, it's even reproducing that information, and some of it mutates and some of it survives. Uh,
Okay. So is it the act of consuming energy and I guess kind of thinking, quote unquote, that constitutes it being lifelike? Or
there are a lot of definitions of life,
okay.
and some definitions of life rely on understanding how information is replicated,
Okay.
and the both the energy and the information loss that occurs during that replication. Uh, it you know Maybe some scientists are familiar with the term RNA world. The idea that RNA can act as both an enzyme, so it can metabolize reactions or can catalyze reactions, and it can also act as an information carrier. And for a very long time, people thought that that was the answer to abiogenesis, that we could just needed to make an RNA, maybe just one RNA, but maybe a group of RNA that could carry information and catalyze their own reproduction, and that that would be sufficient for a living system. And so, if you think about that and how that applies to a computer program, right, that's
Okay.
pretty translatable. That a computer program has a lot of those same properties. It can maybe replicate itself, maybe it can change its information based on its environment. For example, you give it new information, and AI can kind of update what it is, what its function is. Um, and yeah, and so I
Okay.
think that there are some really good analogs between what we see in silica and what we see in vivo.
Okay. Yeah, because I mean, I guess if you if we pull the thread a bit, if you talk to the AI, the AI purists, they some of them would say that, artificial intelligence isn't really intelligent, right? It's just the regurgitation of human knowledge and synthesizing it in a new way, or giving you a different answer, but it's basically it needed all this information in order for it to be able to generate this new content. Case in point, like programs like ChatGPT or this new DeepSeek that just came out of China last week, right? So, but then I guess taking your point, a baby learns based off of information that its mother or father or the world or its environment is provides it, and it learns from that information. So I guess it's kind of similar.
Yeah, well, and I think that the publicly available AIs are not. the most advanced AIs that are available, right? So maybe your summary of what an AI does is, very much what we get free from, OpenAI or Gemini or even Copilot, right? But the most advanced AIs are really capable of writing their own code. They're capable of modifying parts of what their protocol is or what they're being asked to do.
Okay.
And so I think it is very advanced, and probably the general public, and of course not even myself, really understand what the most advanced AIs have been asked to do. But yeah, we see like the real infancy of AI, the publicly available one.
Okay. Yeah, because I guess if you're talking about an artificial intelligence like writing its own code, would that be equivalent to a genetic mutation in a human being? Because I mean, we can't really change our own code, right? Or am
Right.
I? Okay. All right.
Well, I mean,
Well,
it's
I mean, without some type of external intervention, I
yeah.
guess, right? Like,
Yeah, there are some interesting things about our genetic code that are modifiable. So epigenetics is the modification. It's basically silencing or activating genes based on environment. And I think that's different than actually like CRISPR, making a CRISPR modified human, right? So, yeah, but I mean, CRISPR modified humans are probably not in our far distant future.
okay. So yeah, so CRISPR would be kind of like the computer rewriting its own code because it's, snipping and making things the way it
Yeah.
hopefully wants them to be, I
Yeah,
guess.
right now we do it the old-fashioned way, which is we select for the most fit individuals in the population.
Okay. Okay. Interesting. Very interesting. Okay, so I guess since we're talking about AI, right? And it seems like to be pretty pretty strong in some of the work that you've been doing. Like, so I guess how has artificial intelligence accelerated the discovery of abiotic vesicles for you, for in your work or in, biochemistry writ large? So,
Yeah, so I think the thing that I'm a big advocate for is for everyone to try out improving their performance using AI. So if there's a take-home message from this, it's that we just need to lean into it because it's a tool that's not going away, and if you are not using it, then you're going to be not as efficient as the people who are using it. Um,
really? I don't know. You might have an we might have an argument about that.
well, so in our work explicitly, we have a lot of big data. So the problem with membranes is that they are very compositionally diverse. Biological membranes, there are hundreds, maybe even thousands of different molecules that make up your lipid bilayers. And when you think about finding a solution to making the most fit individuals out of all of those different compositions, it becomes really, really challenging. So that was our goal. We have a big compositional space. We didn't even start with like everything we could have. We started with a kind of small subset. Only 7 different amphiphiles were used in our study. And even that, in 7-dimensional space where we are setting limits on compositions, it is a large space to understand. Most experiments up until this point have used only 1 or 2 different amphiphiles in their research. Some have used up to 3, and when you are thinking about a composition of just 2 molecules, how do you decide what ratio even to use? So most then scientists would use maybe 1 or 2 ratios to try to understand the fitness of these abiotic vesicles. Well, it turns out that if you want to work more efficiently with large data, then applying some AI to your analysis and having the AI like recommend your next set of experiments is going to be a little bit more predictive than a graduate student I don't know, like looking at a micrograph, I guess, is how we did it in the old days. And so we have computer processing our micrograph images. So that's not AI, that's just basic programming code, but it is really, really effective at, we have, every week we would have, I think it's like 500 images that we would need to count all of the little circles in. And so it would take our computer code maybe an hour to do that. No, I don't even think an hour, like 30 minutes to process all the images. And if I was a grad student counting all those little circles, it would take me, days. And I would be imperfect. I would be very imperfect at it. And so, sure, our computer program is sometimes imperfect, but it is regularly very, very accurate. And I think that has been a great application of just, using a computer to process information. But then we have this computer output, which we can feed into our statistical analysis. And so I'm not going to go into the nitty gritty. One, it's actually not my field. I have a collaborator, Joshua Schreier at Fordham University, who does all of our computational work, along with his postdoc, Hao Lu. But we essentially use a Gaussian model to predict whether or not a composition will form membranes. And we did 6 rounds of prediction, and then in the last round we said, hey, like, we think our model is really good. We've been working in pretty high concentration ranges, and so we asked the model to cut our concentration from about 6 millimolar to less than 3 millimolar. So this is a very low concentration of molecules in solution. And it made a prediction of 24 compositions that should form membranes, and all of them actually formed membranes. So it was good at even extrapolating the previous data. So this is, a statistical analysis of our data. Maybe you might call it machine learning instead of artificial intelligence, but it was, we put in data into the model, and the model predicted correctly compositions that would form very low, form membranes at very low composition or concentrations.
Sarah, so can you tell me more about how you visualize these vesicles then?
Yeah, so they're very tiny. Most of the structures that we're looking at are maybe between 100 nanometers to 100 microns. So when you think about a E. coli, and E. coli is about 1 micron, so many of our structures are about that size. But we do see much bigger structures that are more similar to mammalian cells, or even bigger. They're so big that you could almost visualize them with the naked eye. They're almost a tenth of a millimeter. But we don't use our naked eye. We actually dye our vesicles with a hydrophobic dye, so it inserts into the bilayer, and then we use fluorescence microscopy to look at them. And at CCSU, we don't have a microscope that performs on this level. They're very, very expensive. So we actually go to the Yukon core facilities to visualize our structures. And we use a confocal microscope. So that's a type of microscope that uses a laser to actually excite the fluorophore. And it's like a very specific wavelength. So we're really honing in on only the molecule that is in our membranes. And then we can actually we put all of our samples into one big plate. So I actually wish I had one in my office right now. And they hold 384 samples, so they're called a 384 well plate. And each little well in the plate has 25 microliters of sample. And then the microscope is a robot. And so we just put the plate on, and it has a computer program that says, look in each well, take 4 pictures in each well, and then move to the next well. And so it just goes zzz, 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 and it moves through the entire plate without any human intervention. So, we send a student there to take the samples, and then they sit there and do homework for an hour while the plate runs, and then they come home with, these 500 micrographs. And so it's a really, really efficient way to have students doing research where, we've automated the microscopy. We've automated actually counting our micrographs or analyzing our micrographs. We've automated the prediction mechanism, right? How do you analyze your data? But then also in our sample preparation methods, we've automated our sample preparation. So we have a liquid handling robot in our lab, and we put in a computer code, and it like dispenses all of our different amphiphile compositions, and then it will also like rehydrate those, and it'll mix them, and then it'll add the fluorescent dye and transfer samples to our 384 well plates. And so we essentially in our lab, we've really taken to not just automating data analysis through machine learning, but also automating the physical work too. So the benefits of a robot are that they're much more accurate, as long as you're using them appropriately. And also you are sure that they haven't made a mistake, right? If a student is pipetting, maybe they put the wrong thing in the wrong well, right? But with the robot, it always just does the code you tell it to, and you can always go back and look at the code and see, oh, did I tell it the wrong thing? And so there's a lot of really cool accountability that you can have when you're doing science, when you are automating things, not just automating the data analysis and using AI to improve your workflow, but also when you're thinking about automating your actual preparation of experiments. And I think that's one of the things that has been really instrumental in moving my research forward, is really embracing these types of technologies so that we can look at harder or less tractable problems, things that maybe people wouldn't have approached in the past because we can only process 10 samples in a day, right? And so we've had the ability to look at thousands of samples in a few months.
Wow. Okay. Yeah. So yeah. I mean, it that definitely makes sense, especially at such a at that small of a level. Details really do matter, right? So any little variance or change could potentially completely screw up or change your results, right?
Yeah.
So consistent. Wow, that's a very good point to bring home for our listeners. So thank you for that. Sounds good to be a. Sounds like you. I would want to be a student, right?
Yeah, and
Get my homework done.
I would also say that our students are really excited about this because it's a great opportunity for education. I think students often think of chemistry as, I don't know, like fuddy-duddy or like a science that people used to do. I've heard students say that. I was like, that's something people used to do.
Oh man.
But there really is a lot of modern techniques that we're using. And, you can now be expected to learn to code, for example, in a chemistry lab, because you're going to need to use Python to analyze your data. We use a lot of statistical analysis, so that's in R. And so there are a lot of opportunities for training that are instrumental in industry now, that you learn with a chemistry degree. So I think it's a really great training opportunity for my students because they learn a lot about robotics and a lot about instrumentation and a lot about computer programming.
Yeah, and those skills are obviously portable to
Yeah.
industry because I mean you could use it, you could continue on in industry, whether you're in academia or you're working for some Fortune 500 company, but in a in a chemistry discipline, but you can it also expounds to other disciplines,
Yeah.
right? So that's pretty profound, like real world experience that your students are getting.
Yeah.
So so I guess to let's if we can try to bring this full circle, like how does this all scale to human existence, right? Like because I mean at the at microscopic or nanoscopic, I mean these small scales that you're speaking of, you know, we humans understand things that's right in front of our face. So how can we let our listeners know like clearly this is important work, but just so that they can understand, the gravity of it all.
Yeah. So there are a lot of fun thought experiments that we do in origins of life research. And I think one that's really profound to me is that pretty much every surface you have ever seen on Earth has been touched by life. We really don't know what it would look like if there wasn't life to significantly modify every surface that we are looking at. And I'm thinking about the suburbs, right, where there's manicured lawns and we have put asphalt on lots of surfaces, right? But also even in places like deserts and where you think, or like the Antarctic, right, where you think that there's not a lot of life there, even those locations have been modified by the organisms that live there. We historically have focused on studying mammalian life because it's easy. They're big and like us and domesticatable sometimes. But there's a lot of life that is really strange and wonderful out there, and we can learn a lot from that type of life. But I think that the really interesting part about understanding abiogenesis is that we're asking, if there is a place that does not have any life, how does life get started? And this may be relevant to exoplanet studies, right? But this also could be relevant if we, for example, wanted to make synthetic biology that could perform tasks that our existing life doesn't perform. And so if we can create synthetic biology, then maybe we have the capability to generate rules for what life is. We could make new novel compounds extremely cheaply. There are both economic benefits and like basic science questions that we can answer with this type of research. And if you think about physics, so physics had a big blossoming when Newtonian physics, so thinking about like gravity pulling an apple out of a tree, became formalized mathematically. And then there was the quantum revolution, where we really started to understand mathematically how atoms or subatomic particles are arranged. And there are very, very specific rules for those arrangements. And so if you think about it, we don't have a really good mathematical interpretation of what life is. And with a second instance of life, whether it is something that I create in my own lab or whether it's something that we find on maybe Mars waters, we will be able to maybe start to generate some mathematical interpretations for rules for understanding what it is required to be a living system.
Okay. Yeah. So okay. Yeah. It's when you were the describing uh, the relevance here. One thing that shot in my mind was, I mean, it's a movie. It's an old movie, but it's Independence Day, and it came out like in the late '90s. And I think for me, the most important part of that, besides, I mean, it was entertaining, right? That's all it really was. You're not going to win an Oscar for it, but the thing that struck home to me was the reason why the Earth was saved was because of small bacteria that took out the aliens because they didn't have an immune system for the bacteria, right? So, when you're speaking of, life at this scale and everything, it's really profound, right? And I thought, that movie was a clever way of, almost kind of speaking about, chemistry, biochemistry in a way, and, pushing forth science in a way that, people didn't really realize that, they should actually understand. So, thank you for that response.
Yeah. I think you have great taste. That's one of my favorite movies. But also, like that's a really good point that bacteria are some of the most flexible, or single-celled organisms are very flexible in the way that they're opportunistic. And I think, thinking about how flexible they are, um, it makes it really tough to think of making a synthetic biology that could like escape the lab. So I've gotten that question a lot, right? Like,
Yeah.
oh, aren't you afraid that you're going to make something that's like going to be deadly to the human race? And the really interesting thing is that most life is highly evolved to live on planet Earth in the exact niche that it is in. And the likelihood that in my lab I create this organism that is so robust that it can outcompete existing life on Earth, it seems like a very low probability event to me. I think we are actually much more worried about, as we explore more of the universe, taking our bacteria to other places. And planetary protection is a serious, serious portion of NASA's mission. They try really hard to decontaminate everything, and even with their best efforts, sometimes when missions return, they can tell that bacteria have been living in those missions without their knowledge. So I think we are. It is likely that bacteria is out there in the universe from Earth.
Okay. Yeah, I mean, just making it, putting it in front of our faces, like it kind of is similar to, when you go abroad and you buy fruit,
Yeah.
right, or some type of produce, like you can't just bring it back to the States, like you have to make sure it goes through customs, because if there's some, disease or pathology or bacteria or something on that fruit, you don't want to bring it back here and cause some harm, or a pesticide or insect or something that causes some harm to our local produce, I guess. Is that kind of similar to an analogy to taking bacteria to other planets, or?
Yeah, I mean, like we know bacteria is really good at adapting to new environments, and or I mean, like, or I think a good example is like rats, right? Like
Hmm.
rats got taken all over the world on ships because they
Sure,
didn't know
yeah.
they were there, right?
Yeah.
And so, if bacteria does find a water phase that has a good source of carbon, it probably is going to try to live in that, um, regardless of what planet it's on.
Okay. Okay. So I guess, if we discover or build another type of life, I guess, how will that change our understanding of biology?
Ah, I think that it depends on how different it is from our existing life. But a really good example is that we assume that water is a universal requirement for life. And we don't actually know that that's true. All life we know has a internal water compartment, and it relies on water for all of its reactions. It would be really interesting to see if it's possible to generate life in a non-water solvent. And that would tell us a lot about the requirements for information, but also a lot of energetic requirements for life that we assume to be true because we are water-based life. I think there's been a lot of discussion about like carbon-based life, and based on I'm a chemist. Like carbon is like a really good molecule to make life out of, and so I think that we'll probably find carbon-based life if we find it. Um, the other thing that we don't really know is, proteins are really good at catalyzing reactions. That's why all of our enzymes in our body are proteins. But it would be really interesting to see what a different catalytic polymer looked like, or a different catalytic mechanism looked like. And that's not directly my work, but we do work on, polymerizing molecules and building out structures and aggregates out of insoluble polymers. And there's some really cool work using polyesters instead of using polypeptides to make structures that are functional. So I think we have some cool synthetic inklings at this point of how to get alternative catalysis from different molecules. And so it'd be nice to confirm that. If we see exactly the same molecules that we are using in our life in another type of life that we're sure didn't come from Earth. It's not bacteria, right? It's a totally different organism. I think that would be so important to understand about how how stringent the requirements for biomolecules are. And so I think that's the key, right? If we get a biological system that looks exactly like our life, then we would know that there's really only one way to do this. And if it looks very, very different than our life, then we can say that there's much more flexibility, and we can probably make some really weird things in the lab with that knowledge.
Okay, so just there something just came to mind, and so you mentioned earlier about NASA doesn't want to contaminate other planets, right? So it's probably a two-part question. So is there life elsewhere, right, in our solar system and universe, right? First one, but also could that life be potentially a result of the contamination of Earth life, but but would we be able to know that it's Earth life because it has fingerprints of Earth, or what if it like evolved in a way, or could it evolve in a way where we wouldn't have an understanding that it actually was Earth life? Does that make sense?
Yeah, yeah. So I think there's no good answer to your question. We do know that on Earth we have found meteorites that are from Mars. So Martian meteorites exist, which means that something hit Mars, it ejected part of the Martian terrain. That rock flew to Earth and landed on Earth, and we have found it. Right? So think about like all the probabilities that are involved in that and how often this much must actually occur.
Sure.
And then the reverse. So how many Terran meteorites are on Mars? And did those meteorites have bacteria on them? Probably, right? Did that bacteria survive space? Maybe, right? Space is like a really extreme environment, but there's a lot of ionizing radiation in space, which makes it hard for living things to survive. But we do know that it's not impossible. So I think Mars, if we find something that looks exactly like Earth life, it's probably Earth life. That's less likely, though, as you get out to the moons of Jupiter and Saturn, just because they're newer, they have a much shorter history. They have very, very different environments in them. That Mars actually had a period of time where it was an Earth analog, so it was wet and had an atmosphere that was very similar to Earth's early atmosphere. So it's possible, both things are possible on Mars. It's possible that Mars evolved very similar life to what we have, just because it had a very similar environment, but it's also possible that we have contaminated Mars. And in fact, it's actually kind of like, I mean, like we've sent enough rovers to Mars that there's a good possibility that there is fingerprints of Earth life on Mars. Not in abundance because it's such a harsh place now, but I think elsewhere in the universe, right? The universe is a really big place, and so there's likely life elsewhere in the universe, or I don't know. There's like this mathematical condition where there's either zero of something, there's one of something, or there's like infinity of something. And we hope that we're not one of something, right? A set of one. We hope we're a set of infinity. But the reality is that most life and most of the history of Earth was single-celled. And so if there is life elsewhere in the universe, most of that life is probably single-celled organisms. And that means that if we can't fly a spaceship there or send a physical object there, it's going to be really hard to detect it. So like we are doing, I don't know if you know this, but we're doing a lot of using the James Webb Space Telescope. We are discovering a lot of exoplanets. We're learning a lot about exoplanets, but there's no good biosignature. There's no agreed upon biosignature, and I think it will be really hard to claim that we have found life on an exoplanet without physically going there, which is like getting to the closest exoplanet is, with our current technology, is lifetimes, I think. So we're pretty much stuck in our own solar system at this point, looking for life.
Well, I guess on the bright side, it is a nice solar system. So.
Yeah, the best, really.
But so just if we can, if you just indulge me a bit, so if just thinking about, maybe we contaminated Mars with some of Earth life. Would it be serendipitous that if we went there and we found the fingerprints of Earth life, but it was still living, is there a way to figure out, oh, well, maybe whatever it did to survive, we can do to survive? Or is that kind of just, I'm just
Oh,
crazy, crazy thinking right now?
that's a really neat idea. I, you know, I think it depends on how recently it has immigrated, right? There's some single-celled organisms that reproduce really, really slowly, and it's possible that they have undergone not very many generations since they arrived on Mars, so they wouldn't have had a lot of time to evolve. Um, but you're right, we might be able to infer things about what has survived that process. If they actually have evolved for a really long time, independently, it's a really, really cool experiment, right? Because if we can like trace back the lineage, then we can look at how something that is so different in such a different environment evolves, and we might actually see things that look very alien in those evolutions because they had to deal with really high perchlorate sample solutions because that's what Mars has, right? And so it might still have evolved very far from what we currently see in life. And I think, from as a biological perspective, that's still a really interesting result, even if it is like panspermia, essentially.
Okay. No, that would be, yeah, that would be really interesting. Very interesting. So so that's definitely something that it's just it's too much to think about. But yeah, thanks for indulging me on that. So I guess, for some of our listeners, uh you know I guess what sparked your interest in becoming a scientist? And if you could, what kind of advice would you give some of our younger listeners that, uh, would want to aspire to become a scientist much like yourself?
Well, I think I first said like I want to be a scientist when I was I'm going to go with kindergarten. When I was in kindergarten, I was like I want to be a scientist. And what really drew me to science was Albert Einstein's hairstyle.
Okay.
I don't know why, but like it was really compelling to me to like just get to have like crazy hair and for people to still think that you're, competent, um, which
Okay.
that is definitely the 5-year-old interpretation of career choice, I think. But my parents always really encouraged me to be curious and to I grew up kind of on a farm, and so we got to explore biology really pretty intimately, with like the birth of animals and chicken eggs and insects and crayfish. So there was a lot of scientific exploration that happened early in my life, and shaped what I wanted to be. I was always really good at chemistry, even in high school. And when I got to college, the origins of life just really piqued my interest. And so I like started on that path really early in my career. I decided I wanted to study the origins of life and figure out how to create life in the lab. That's always been my passion. I like hard problems, I guess. And I think that one of the things that maybe draws our students in is that you really do get to work on things that are hard problems that are very basic science, but that have real applications, real benefits to society. And that's the great thing about becoming a scientist. I think that one of the challenges that we face in recruitment, essentially, in convincing students that this is a career path, is that it doesn't look as flashy as other types of jobs. It is if you're a very social person, then you have to make sure that you build social interaction into your day to day, because sitting in a lab for 8 hours is not very socially stimulating. But scientists, some people think of scientists as like these very like quiet geniuses or isolated perfectionists or something like that. And that is definitely not my experience. There's room for everyone in science, and we need lots of different personality types. And you know, modern academic labs are not these monoliths of white lab coats and perfection. I think we're really working towards, solving hard problems in interdisciplinary teams. We're really focused on building infrastructure and making sure that we have support for the kinds of questions we want to solve. And we're also really invested in training the next generation of scientists to think critically and be members of the public and go out and promote science in their local communities, right? And that's important too, right? We need an educated public that understands how to interpret data and how to really fact check things. And I feel like if anything, it's important to get a science education. Even if you're not going to be a scientist, you can apply those principles to a large number of other jobs. And we kind of touched on that, but now in science we have all of these instruments that generate huge amounts of data. And how do you ask questions of that data is one of the challenges that we're going to face in the future. So data science is at the forefront of a lot of chemistry now. And so I think that students get a really good critical understanding of modern problems that we're having in our society. We also get the chance to network and go to conferences and talk about things that are important to us. We get the chance to do things like this and talk to fun new people, and create opportunities to learn about our work. I just think that I have 3 children, and I hope at least one of them is like, oh yeah, science is my jam, because it's just like, it's such a great place to be. And I really enjoy working in chemistry in Origins of Life.
Yeah, no, it definitely comes across that you're passionate about, your work, and fundamentally what you're doing is necessary, for our fuller understanding of who we are, right, as a species. So really, thank you for what you're doing, and you know, uh, what you're doing is an inspiration to a lot of our listeners, and thank you for giving them a bit about how you got your start, because that allows them to identify with somebody, and that's important in order for people to try to take that next step. So really appreciate that.
Thanks.
So on behalf of all of us living in Connecticut and others tuning in from other states, thank you, Sarah, for sharing with us information about accelerating the discovery of abiotic vesicles. You've given us a lot to think about. Listeners, I encourage you to subscribe to this podcast on Apple Podcasts, Spotify, Amazon Music, or YouTube, and visit the Academy's website at ctcase.org to learn more about our guests, read the episode transcript, and access additional resources, as well as to sign up for the Case Bulletin. Thanks again, Sarah. This has been a fantastic opportunity to talk to you.
Thanks, Pam.